Hello, we will now continue on with more parts of the neuron, what they do. We'll talk about the synapse, the cytoskeleton, and the plasma membrane in this one. So the synapse, a lot of action goes on here. Synapse is where one neuron influences another neuron. It's the space between them. And when we say one neuron influences another, we often say it innervates. So when you hear me say the word it innervates, innervation, that is when this neuron here is influencing this second neuron here. Now the first neuron, here we have the axon from the first neuron, the axon terminals, here is in red, and the axon terminal is going to spit a neurotransmitter onto the second neuron. The first neuron is called the presynaptic neuron. The second neuron is called the postsynaptic neuron, and the space in between is called the synapse. So if you look here in red, these little, this axon terminal here, that would be a close-up of the axon terminal of the presynaptic neuron. And here it's influencing the dendrites of the postsynaptic neuron. So in this postsynaptic neuron, this will be on the dendrite. And there's little receptors. So we'll spend a lot more time on them later in the semester, but the receptors are on the postsynaptic neuron, and in the presynaptic neuron there in red, we have vesicles full of neurotransmitter molecules. And we also have a lot of mitochondria there. What else? There's no Nissl bodies, no rough endoplasmic reticulum in the axons probably. We probably won't talk about it because we're still learning that. And the axon membrane is different from the membrane of the dendrites and the somas because they have more voltage gated channels. We'll talk about that. All right, so you have the basics of the synapse, the presynaptic and postsynaptic neuron, and the space in between is called the synapse. So let's talk about the cytoskeletal elements. So what are the components and what do they do and what are the differences? So just big picture. We have microfilaments. These are the smallest, and I'll explain what they do. They do things like changing the shape of the cell. Neurofilaments, those are the medium size, and those are for strength, and then microtubules are the largest. So for here, I just arrange them in order of size, and then I'm going to spend a second to tell you what they do. So let me go back to the microfilaments. Microfilaments, these are the smallest cytoskeletal elements, and that might be things like they're located near dendritic spines. They change the shape of the cell. They help with myelination. So those are the microfilaments. Neurofilaments, these are the most common. These are going to give strength and structural rigidity, as well as, these are going to give support and strength. The rigidity we'll get to. These give structural support and strength to the axon. These are the most common ones. But the ones I'm going to spend time with are the microtubules. These are the largest cytoskeletal elements, and these are for structure down the axon and for movement. So let's spend some time on these. What a microtubule is, it's a hollow tube, and it runs down the axon. And so they help give rigidity to the axon, support, and they help transport materials. And the transport can go in two directions. So there's anterograde and retrograde transmission. So anterograde is going to go from the soma down to the axon terminal. So that might be like neurotransmitters. There are some neurotransmitters that are synthesized in the cell body, and you transport them down to the axon terminal. There's also retrograde transmission, and that goes the opposite way. That's going to go from the axon terminal back up to the cell body. And that's going to be stuff that you need to break down. Stuff that needs to be degraded from the axon. So, and it also is going to give signals to the cell body about metabolic needs. So transport goes in two directions, and it's also some is slow and some is fast. I'll get to that. So how things are transported. You enclose material in the vesicles, and these vesicles kind of walk down the microtubules. There's dynein is a protein that's kind of the legs. Oh, and this is the direction of transport. So there's anterograde that goes from the cell body down to the axon, because As far as we knew, until recently, you couldn't create a new protein there in the axon. There wasn't any rough endoplasmic reticulum or ribosome. So we thought, oh, we have to make all proteins in the soma. But now that may not be true. Neuroscience, we keep learning new things. But generally, we synthesize the proteins in the cell body and move them anterograde down to the axon terminal. But retrograde goes backwards from this axon terminal to the cell body. And it's some stuff that it needs to degrade, and so it gives some signals to the cell body about metabolic needs. Now there's both slow and fast axoplasmic transport. The slow transport, that is going to be enzymes, structural proteins that you need for small neurotransmitters. The small neurotransmitters, those are synthesized near the axon terminal, and the large ones are actually synthesized in the cell body. We'll get to that later in the semester. But the slow is movement of about 1 to 10 millimeters a day. And now some axons are a meter long, so that would be really inconvenient. It would take 6 months or something to get down there. There's also fast axoplasmic transport, which is up to a meter a day. That's going to be more membrane-bound materials, synaptic vesicles, membrane proteins, peptide neurotransmitters. I'm mentioning this. Mention this because with the microtubules, there's something called microtubule-associated proteins. And these are important in helping microtubules anchor to each other, and they may be abnormal in Alzheimer's disease. There's an axonal microtubule-associated protein that detaches and it collects in the body of the neuron and can cause problems. So I'm mentioning the whole cytoskeletal elements mostly for those of you who might be interested in Alzheimer's disease and you want to research that. But that's all I'm going to say about them. All right, they're important. The microtubules are important for structure, rigidity of the axon, transport of materials, and materials can go in both directions. The end. We're good. All right, what I'm going to close out this video with is the plasma membrane. All right, stuff we need to know about the plasma membrane to understand how action potentials happen. And the plasma membrane, we all learned about the fluid mosaic model. And the plasma membrane has a lipid bilayer. Remember those phospholipids and cholesterol, and they're not directly bound to each other, but it's nice and fluid and flexible, and so it doesn't crack. And there's these mosaic proteins that are shoved in randomly around the lipid bilayer, and that's for selective transfer of substances through the lipid bilayer, because some things easily cross the lipid bilayer, so small nonpolar molecules, but things like glucose can't cross the lipid bilayer. To get glucose into a cell, you need a protein carrier. To get certain substances, you need a protein. And there's a lot of different types of proteins. There are carriers. That's going to be a specific transport of substances across the membrane. That's, for instance, the sodium-potassium pump. So if you've taken physiology, you remember it. If you've taken cell bio, you should have covered that. So a carrier protein, for instance, the sodium-potassium pump, is going to pick up 3 sodium and pump them out of the cell, and 2 potassium are pumped in. So it's going to specifically transport specific substances across the membrane. There's also receptors. Receptors we'll talk a lot about when we get to drugs. Receptors have a specific size and shape. They're a protein. And if you are the right size and shape that binds to that protein, great. You're going to bind and start a sequence of events. So for example, there are acetylcholine receptors on your dendrites in some areas of the brain. And if acetylcholine binds to those receptors, it opens up a sodium channel. Sodium comes in. All right, and other ones, if dopamine binds to other areas, it starts another sequence of events that's going to lead to a second messenger and a protein kinase being activated. So something binds to the receptor. Aldosterone binds to a receptor. It's going to get into the nucleus and start creation of a protein in the kidney. So things bind to the receptors and start a sequence of events. All right. So receptors are important proteins on the membrane. Then there are channels. Different types of channels. There are passive channels. Sometimes we call those pores. So that is just going to be a protein pore. It's always open. It's passive. You can think of it. You can think of a channel as a protein that it spans the membrane. You could think of it as a bridge across a river. All right. So this passive pore, this passive channel, is a bridge that crosses the river. There's nothing saying you can't cross it as long as you're the right size and shape. So if I have a little tiny bridge, an elephant can't cross it. So if I have a passive channel for sodium, something larger than sodium can't get through the sodium passive pore. But if you're the right electrical charge, you're the right size, yeah, it's always open. There's no gate on this bridge. There's no drawbridge on my bridge here. So a passive channel is just a bridge across the river. If you're the right size, you can cross it back and forth as much as you want. But there are other active channels. Active channels have gates. So you can no longer just cross my bridge across the river. You have to pay a fee, or you have to, turn on the button that opens the drawbridge. I hope this analogy is working for you. Okay, so the active channels. Think of a pore. It's just an open channel, but there's a door across it. There's a gate. And some are voltage gated, some are chemical gated, and some are mechanically gated. What I mean by this, a voltage gated channel, if you took physiology, you remember this, I hope, is closed at rest, but when it hits the magic voltage, if it hits the voltage that opens up the channel, the gate opens, and now sodium, for instance, can get on. So a voltage gated sodium channel is closed at minus 70 millivolts, closed at rest. But when you hit the magic voltage, which is minus 55, it's like the key in the lock, whee, that opens up the gate, sodium can rush in. It's voltage gated, so it's going to be closed until you hit the right voltage. And we find a lot of voltage gated channels at the nodes of Ranvier and the axon hillock, because these are important in making action potentials. All right, another type of gated channel, active channel, is called a chemically gated channel. Very similar thing. It's an ion channel, for instance, a sodium channel, that is closed unless the right key goes in. So if acetylcholine binds to this channel, it opens up, sodium comes in. If, let's see, Xanax, if Xanax binds to a GABA channel, it opens up and chloride comes in and makes it negatively charged. So a magic chemical has to bind to this kind of channel, and that will open up the channel then. So those are chemically gated. They'll be opened when the right chemical binds. And still others are mechanically gated. So when the right movement happens, they open up the channel. We'll talk about those later. That happens in the ear for sound transmission. So voltage gated channels are opened when you hit the right voltage. Chemically gated channels are open when the right chemical binds to them. And we'll see these in dendrites in the cell body. And mechanically gated channels open when you literally tug on something to physically open up a channel. So just to review these, this table isn't in your notes because I just decided to add it, but you can just jot this down at the bottom of your notes. So when we're talking about channels, there are passive channels that are just pores. They're always open, and if you're the right size and shape, you can just fit on through. If you're too big, you can't get through. If you're the wrong electrical charge, you can't get through. So it's a bridge over a river. A person can walk across it, but an elephant can't. But they're always open. There's no fee. There's no drawbridge. There's no button you have to push. And passive channels, these are located all over. The pores, the passive pores are all over the neuron. And they're really important in the resting membrane potential, which we'll talk about in a couple videos. Chemical gated channels, these are the ones that will only open if the right chemical binds. So if acetylcholine binds, great, we've opened up a sodium channel. All right, and these are located in the dendrites and the cell bodies. Because if you think about it, a chemical, a neurotransmitter, is going to be released from the axon terminal and bind to the dendrites. And so when the chemical binds to the receptors on the dendrites, it opens up an ion channel. And these are going to be involved with something called synaptic potential, we'll talk about later. And finally, voltage-gated channels. These open up when you hit the magic voltage, which in neurons is about -55. So these are important in making action potentials. And those are going to be in the axon. They're going to be in the nodes around VA and the axon hillock. And I'll just close it out by saying we classify neurons in a lot of different ways. We already talked about this. Some we classify as sensory or afferent, motor or efferent, or interneurons. Sometimes we classify neurons based on what neurotransmitter they release. So we call some neurons a GABAergic or a dopaminergic neuron. And some we classify by how many extensions from the cell body. The ones we're going to talk about are multipolar, just that one axon. But later we're going to talk about bipolar ones when we get to the senses, when we talk about the eye. And I'm going to close this one out for now. In the next video, we'll talk about neuroglia, and then we'll start to get into the physiology of it. Alrighty.